OK， 现在我在 record 了。OK, Rooney, I think maybe you can start from now.
可以了啊。OK， 大家好，时间差不多，我们就开始了。不好意思，今天推迟了10分钟，一些 technical 的 issue。首先自我介绍一下，我是高如丽，是美国西北大学的一名助理教授。我们的科研主要是利用单细胞组学的方法进行肿瘤进化研究。最近有一些课题延伸到心脏移植、慢性排异反应的领域。但是言归正传，今天的主角呢是我们加州大学洛杉矶分校的李敬义教授。然后今天的主题呢是庆祝他荣获2023年这个 Overton Prize， 这个是一个大奖啊。这个 Overton Prize 是2001年国际计算生物学学会 ISCB 为了纪念那个杰出的生物信息学家 Christine Overton 而设立的。这个奖呢，用于奖励在生物信学领域获得杰出成果的，主要是年轻的科学家。这个奖每年颁发一次，每年呢只奖励1位科学家，只有1位获奖者，所以是很不容易的，很 competitive 的。往年的获奖者可以简单列一下，包括呢 Christopher Berger、David Baker、V. Regev、Dana Peer、Kerr Chapnell。还有另外一位华人啊，是那个伊利诺伊大学的香槟分校的建鹏，这些都是鼎鼎大名的计算生物学家，还有其他的一些 list 有22位，我就不一一列举了。我们这个李敬义教授获得这个就是这个奖励，真是实至名归，因为他在这个生物学和统计学交叉领域做出了很重要的贡献。然后上个月呢，就是6月份，2023年6月份，就是国际计算生物学学会 ISCB 3位作者 Christina、Daniel、Martin 他们呢专门写了一篇文章，报道了他这个获奖的经历以及他个人的成长经历，都在上面发表在 Bioinformatics 杂志上面。李教授是2007年获得清华大学生物科学本科学位，然后2013年从加州大学伯克利分校获得统计学博士学位，师从Dr. Peter Baker and 海燕华2位教授。就是我数了一下，李教授从13年博士毕业到现在，只用了10年了，从博士毕业到那个现在是 full professor 正教授。然后呢，给个参考，我自己是2015年博士毕业的，我现在是助理教授。然后数了一下李教授的简历，就是从13年到现在，大概有20个各种奖项，就是非常了不起。然后李教授这个跨学科的经历，对我们就是无数跨学科、跨专业的小伙伴们来说呢，是个巨大的鼓励。所以我今天很荣幸能就是说 host 他这个讲座。好，废话不多说吧，然后 science 的部分就是正经的 science 还是交给李教授来讲。就是今天非常感谢李教授抽出宝贵的时间给我们就是讲解数据分析当中的一些统计学的原理，以及呢他最近刚刚发表在 Nature Biotechnology 上面的单细胞数据模拟的工具，已经到第3代了 IC Design 3。所以下面就有请李教授吧。
Many thanks to Dr. Ruli Gao for your very generous introduction, and also many thanks to Dr. Kai-Fu Chen for hosting this event for me. It's my great honor to present my work before I will give this formal presentation in about 3 days in the conference. So the work today is an ensemble of my recent work, which are all under this theme of using synthetic controls to enhance statistical rigor in genomics. So I should first introduce the logo from my group. I title my group as the junction of statistics and biology. And a very important reason why I came up with this name, thanks to my collaborator, Dr. Xin Tong's suggestion, is that my last name is so common. So calling my lab as Lee Lab is not really distinct. So the name actually reflects our research focus. We want to build a junction of two fields. And as my slides goes, you will see that I use those three colors to refer to three meanings. So when it's something related to statistics or rigor, I use the cyan color. When it's related to biology or scientific questions, I use the green color. When it's about our proposals, it's about this purple color. So first, my motivation for doing this synthetic control is from my biological experience, which is in every experiment, we need to have a negative control to really be sure that our findings are real. So in this cartoon from Khan Academy, we see that to evaluate the effect of water on tree growth, we need to have a negative control group in which we didn't give any water. And then we compare the resulting growth, then we can see, yes, the water makes an effect. So this is what we need in experiments. And you can see that here, the control group is a concrete object. And in data analysis, in statistics, a commonly used concept is the null hypothesis. I found this concept definition from this awesome fintech to be very accurate. It says a null hypothesis is a type of conjecture used in statistics that proposes that there's no difference between certain characteristics of a population or data generating process. So you can see it's a conjecture, which is an abstract concept. So what I want to emphasize here is that in our data analysis, when we do statistics, the null hypothesis should not depend on the test procedure, how we do the data analysis, or the data itself. It's just what we have in mind. So furthermore, what is hypothesis testing given the null? It will provide a method to reject a null hypothesis within a certain confidence level. What's in the parenthesis is very important. Null hypothesis cannot be proven though. So you see that it's something in our mind and we cannot say it's right or wrong just by doing statistical analysis. We can only have a conclusion, say rejection or non-rejection at a confidence level. However, when I search for null hypothesis, the top hits that Google give me is different. So here's the message I want everybody to bear in mind is that our data is random. So with this in mind, we know statistics cannot tell us everything. But when I do Google search, we can see that these two top hits are not accurate. The first one says, a hypothesis, null hypothesis is a hypothesis that proposes that no statistical significance exists. But as I said, the null hypothesis should just be a conjecture. It shouldn't depend on our procedure. So this is a misconception. On the right, it says a null hypothesis is deemed true until proven wrong based on experimental data. But we just said it cannot be proven wrong by data analysis. Therefore, you can see how common this misconception is, even though Statistics 101 has been taught in every university to almost every STEM major student. So besides those common misconceptions, I want to talk about three questions. The first question is, what is a proper null hypothesis? And different null hypotheses may lead to different discoveries. I will use examples to demonstrate this. Second, how to make an abstract null hypothesis concrete. To achieve this, I will propose the concept called synthetic null. Third, how to use synthetic null to reduce false discoveries. For this, I will propose a contrast strategy. So first question, what is a proper null hypothesis? I will demonstrate this using a recent paper we published about the bulk RNA sequencing differential expression analysis. So this analysis is commonly performed in most bioinformatics. If you want to decide which genes have expression changed between two conditions, you do RNA sequencing and you collect expression levels of thousands of genes altogether. So in this cartoon, we assume that we have 3 replicates from each condition as 3 RNA-seq samples. Every column here is a gene. So the differential expression or DE analysis is essentially to compare each gene's expression levels under the 2 conditions to see if the difference is big enough so we can reject a null hypothesis that is, the gene is not differentially expressed. If we reject the null, the gene will be called a DE gene. So we are doing one hypothesis test per gene. So for this test, the two most popular methods are DESeq2 and EdgeR. They have been very widely cited, and I just checked that from last year to this year, each method has more than like 7,000 citations. So you can see how often they are used. And their common statistical assumption is that every gene's counts, let's say the 3 counts under each condition, follows a negative binomial distribution, subject to some factor adjustment. So specifically, we assume that the condition 1 has m replicates. So a gene has m counts, and these counts are assumed to be independently following a negative binomial distribution for each gene. And here we would have a mu1 for the common mean parameter of condition 1. Si stands for the size factor of sample I. Similarly, under condition 2, we would have y1 to ym as the counts. So the null hypothesis in both methods here is that mu1 is equal to mu2. But this null hypothesis is only proper when the negative binomial assumption is reasonable. But since the popularity has been there, many people apply these two methods without checking the assumption. And especially, they would apply the methods to non-replicate data. Like in this data set from a Cell paper, the two conditions are 2 groups of patients, 51 pre-treatment, 58 on-treatment patients from this anti-PD-1 immunotherapy. So here, the patients are not replicates. So when you apply these two methods to the data, the red dots indicate the number of DE genes they found. But given the large sample sizes here, 51 and 58, we could randomly permute or shuffle these two groups to create permuted data. But surprisingly, these two methods frequently found more DE genes than they found from the original data. So the bar shows the average across permutations, and the bar shows the standard deviation. Why is this so surprising? Because we shouldn't expect DE genes to be existing in permuted data. So our first guess is that the negative assumption may be the fault. So checking whether the negative assumption holds well in each group of patients. So we can see that this is one gene, and this is what the model, negative binomial model, fits for this particular gene in EdgeR. So we can see that in both groups, there is a clear violation from the negative binomial based on the QQ plot. Because if it fits well, the QQ, the dots should lie on the straight line, but we see clear deviations in the tail. And DESeq2, the same, and actually even worse. If you look at the right group, it's very deviated. And if we look at all the genes together, and we focus on the genes that are rarely identified from the permuted data, the genes that are frequently identified from the permuted data, we check how well does the negative binomial fit to each group. Then here, the y-axis shows negative log of goodness of fit p-value. So the larger the y, the poorer the fitting. So we can see that the second group, the genes that are frequently identified from permuted data, has poorer fitting, as our expectation says. So basically, negative binomial fitting explains why some genes are frequently identified. So maybe some people will tell me, I will do functional analysis for the DE genes, such as gene ontology analysis. And these analyses can give me information for me to decide whether the genes are reasonable or not. But unfortunately, it may not always work. Like in this case, if we take the DE genes found from DESeq2 and EdgeR, and many of them are also found from permuted data, you run the gene ontology analysis to find what functional terms are associated with those genes. You can see that many of the terms are related to immune responses. If you trust the result, you would guess, yes, I found a very interesting discovery, right? There are immune response differences between pre-treatment patients, on-treatment patients. But now you know that these genes are also frequently found from permuted data. You may doubt this conclusion. So in our analysis, we also included a classical Wilcoxon rank sum test just for comparison. The reason we include this is because it doesn't have any distributional assumption. It's a so-called non-parametric test. You don't assume the data to follow a certain distribution like negative binomial. And to be specific, why does the Wilcoxon find no D genes? Very different from the DESeq2 or HR result. The ultimate reason is that it has a different null hypothesis. So I've simplified my discussion here by saying that now I assume the counts are normalized. For example, the normalization can be done by HR. It has a normalization procedure. After normalization, I would have the counts comparable. I don't need to worry about the sample size factor. Then the null hypothesis of Wilcoxon is roughly that if I randomly pick a count from condition 1 and a count from condition 2 for the same gene, then which one is bigger is completely random. So I cannot really distinguish the two based on the relative magnitude. So this null hypothesis doesn't depend on the negative binomial assumption. So we have this paper that discussed this issue more extensively than I just said, and we have more real data sets in this paper. But of course, you could imagine that after we published this paper, a lot of online discussion were stimulated because these two methods were so popular. And we have a lot of following discussion on Twitter. If you are interested, you can check it out. And for this work, I want to thank my collaborator, Dr. Wei Li at UC Irvine, and his former postdoc, Yu Mei, and my student, Xin Zhou. So for that example, the next question you may wonder is, which null hypothesis is more proper, the DESeq2 HR one or the Wilcoxon one? Is this a too abstract question for biologists? I would assume so. But why would so many people trust our results? Here's the intuition. The DE genes found from permitted data are not trustworthy. That's our intuition. But why is that? It's because of our common sense. So then what does permutation provide? It actually provides synthetic null. This is the simplest example of the synthetic null I want to propose. In other words, you may call it in silico negative control because it is a negative control. You don't expect any DE genes to be found from it, and it's in silico because we just obtained it from computer, not by experiments. My second question is how to make an abstract null hypothesis concrete. Synthetic null, I already brought up that idea. And I want to say that synthetic control method is not new. It's already on Wikipedia. It is a statistical method used to evaluate the effect of an intervention in comparative case studies. So basically, it's in the causal inference setting. But what I want to propose here, the synthetic null, is not restricted to causal inference. As I will show you in the following two examples, I basically want the synthetic null to have the association you are looking for disrupted, and the association may not be causality. So I want to say that what we propose here is more general and goes beyond causal inference. So I want to show you another example where permutation helps. So this example is about this common problem about judging the t-SNE UMAP embeddings commonly used in the single cell field. So there is a famous blog about how to use t-SNE. And in this blog, there are many figures showing that t-SNE has a hyperparameter called perplexity. For the same data, you adjust the perplexity value and the visualization or the embeddings will be very different. And so in this blog, several issues were discussed. And here I just highlight two issues. The hyperparameter matters, and the distance between clusters might not mean anything. Like here on the right, they're mixed, right? And here they are very well separated. And furthermore, there is a very famous paper which questioned the use of t-SNE and UMAP embeddings in single cell genomics, right? It says that trying to find an all-in-one visual may be something not achievable. But unfortunately, biologists still like visualizations. So telling them not to use t-SNE or UMAP doesn't seem feasible to me. Then what do we do? So to address this task, we want to formulate this question as, can we decide if a cell's embedding is dubious, which means not so trustworthy, or trustworthy. And our idea is to examine a cell's neighbors before the embedding and after the embedding. So our method for this task is called SED. It is a collaboration with Professor Lucy Xia at Hong Kong University of Science and Technology and my student, Christy. So our intuition is, first, if a cell, we obtain its neighbors in the post-embedding two-dimensional embedding space by Euclidean distance. We also obtain the cell's neighbors in the pre-embedding space, which is usually the principal component space because the cell's original dimensions, which is the number of genes, too high. So Euclidean distance in such a high dimensional space is known to be not so meaningful. So normally, before people run t-SNE or UMAP, they would do PCA on the original matrix and obtain 20 to 50 PCs and go from there. So here, the pre-embedding neighbors means the Euclidean distance neighbors in the PC space. So given those two sets of neighbors, in this case, the cell's neighbors in the post-embedding and pre-embedding space are very similar. So then we would expect the cell's embedding to be trustworthy. So what we just measure every neighbor's distance to the cell in each set. And the distance is based on the two-dimensional Euclidean distance in this space, what I'm plotting here. So I will get two distance vectors between this given cell and its neighbors. So the neighbor whose size is to be determined, that's the only parameter in SED. By default, we set the parameter to be half of its neighbors because we want to capture the mid-range distance. Then the reliability score, we define it as the correlation, Pearson correlation between those two. It reflects how consistent are the cell's neighbors before embedding and after embedding. This is a trustworthy cell. And this is another example, a dubious cell embedding. So here we want to show that the cell's neighbors after embedding are these, aligned with our visual, and pre-embedding, the neighbors are scattered everywhere. So in this case, the reliability score would be low. But then the question is, how can we know what would be a null distribution of reliability scores so we can say, oh, this is low, that was high. So to achieve this, we use permutation. The idea is that for the null hypothesis, here is a cell's neighbors are random after embedding. So basically, after embedding, the cell's neighbors can be any half of all the cells to be randomly assigned. So permuted data will give us such a scenario because permutation the permutation is done independently for every gene. So after permutation, a cell shouldn't be neighbor to any cells. So their relationships become all equivalent, randomly shuffled. So in this case, for each cell in the permuted data, its neighbor should be random, and we can still compute the reliability score. And then for every cell, we will get one null reliability score. Pooling the cells together, we will get a null distribution of reliability scores. So from this distribution, we can say that if a real cell's reliability score is in the lower end, it should be called dubious. If it's in the upper end, it should be called trustworthy. So you can see that here the null hypothesis and the resulting null distribution from permutation serve as a reference. So I'll just show quickly one result. This is a data set from the Science paper, which is the first single-cell RNA sequencing data set of Hydra, which is a freshwater species. And this is the original paper's t-SNE. And using our SED to run it on this data set, we actually detected a few clusters as dubious, which means that their positions in the t-SNE are not trustworthy. And we can also use the SED to optimize the hyperparameter so that the number of dubious embeddings is minimized. So here I'm just showing one example showing what's changed. So before we do the optimization, you can see these three clusters, 2 green, 1 blue, they were scattered, but after the optimization, they were unified into one piece. And our optimized embedding is better or is more consistent with the the gene expression original data, the heat map data. So you see, they are not that different. They're actually very similar, and they are more similar, more different from this orange chunk. But in the original embedding, you cannot see this difference. So that was my example too about what permutation can do. But I would say that permutation cannot always do the job. And there are some synthetic null generation needs that goes beyond permutation. So for the third and last example, this single cell post-clustering DE analysis problem. So this is a Wikipedia figure about what is single cell RNA sequencing. So ignoring the experimental details, basically we are now able to measure every cell's gene expression. So the data we obtain after experiments and pre-processing is this count matrix. Here the rows are genes, columns are cells. So the idea is to identify potential cell types from the data via the use of clustering. And after we obtain cell clusters, the next step is to perform this differential expression, just as I said for bulk data, so that we can know which genes are more highly expressed in this cell cluster as a way to understand the cell cluster and give it a name. So this is the step clustering followed by DE is commonly performed in single cell RNA sequencing data analysis. But this problem from a statistical point of view is double dipping. The same data are used twice. The first step, clustering, would define cell clusters based on gene expression data, that count matrix. The second step, DE, would test if every gene has the same mean expression between cell clusters using the same data. So in this toy example, you can see that the cells are quite homogeneous. It looks like one cluster. But running a clustering algorithm, it could divide the cells into two clusters once you set the tuning parameter to favor clusters. And as a result, both genes would be called differential expressed or DE. But is this DE result really what you want? If you want to annotate cell types, then probably this result is not what you want because we don't seem to have two cell types here. So why would there be such inflated false discoveries? If you think deeply in the statistical formulation, here we have two different null hypotheses. The first one is that, so I would say the ideal one first. So suppose that we look at the M genes in total. So M could be in 1,000 in the data. So we call the M genes expression level the random variable as Y1 to Yn. So each random variable can take one value in one cell. That's the formulation. And we have a cell type, a latent variable. So for simplicity, we consider two cell types. So the latent variable is binary, 0 or 1. But we don't observe this. Otherwise, the problem is solved. And the clusters, which is also binary because we hope the clusters can recover or review the cell types, they are based on Y1 to we denote it hat because it's observed, right? And it's based on our data, it's not the truth. So the ideal null hypothesis should be to compare these two conditional means. Given Z being 0 or 1, does gene J's YJ have the same expectations? That's the ideal null hypothesis. But unfortunately, given we cannot observe Z, we can test this. The post-clustering followed by DE, this double dipping null hypothesis is actually this one. We are conditioning on. That's why we denote this as dipping null. It's not the same null hypothesis. Code, but real ideal null holds. We would have a conceptual false positive cell type marker gene. Because this gene shouldn't be a cell type marker gene, but we reject this post-double dipping, post-clustering double dipping null, then we will call it a D gene. So that's the conceptual gap between the two null hypotheses. Okay, so then the question is, in this case, can we set up a meaningful negative example for discovering trustworthy cell types and trustworthy cell type marker genes? So here the null, in our opinion, is that all cells should belong to one cell type. And is there such a null model for one cell type? For this, we want to use our simulator to help us simulate or generate the synthetic null data. And as Professor Ruli Gao mentioned, this is our recent version of the simulator, SCD-3. And for this one cell type generation, and this work led by my student Dongyuan Song here was based on my earlier work led by my other student Tian Yusun about the SCD-2, the second version. So when the cells are in separate discrete types, these two simulators are actually equivalent. So specifically, let me show you the result, what the null data we have in mind. So in this result, when the data we want to mimic, right, the target data, which is the data we want to analyze, when it just has one cell type, then we want the synthetic null to be like it. And we want every gene to have its mean and variance to be close to what's in the target data. And we also want the gene-gene correlations across all cells to be like in target data. This is one case. In the second case, when the target data does have two cell types like here, what we want to achieve is that the synthetic null data should have the gap filled in. But in the meantime, we want the gene mean and variance to be still preserved and gene-gene correlation to still be preserved across all cells. So how can we achieve this? We use a technique called Gaussian copula, which is used in our simulator, SCD-2 and 3. So specifically, given the target data, we have a marginal model for every gene. And specifically for the single cell RNA-seq data with the popular technologies like 10x genomics, we have empirical evidence to show that for every gene, if it has only one cell type, then negative binomial is a reasonable distribution. So multiple papers have shown this. So therefore, we assume that in the one cell type model, every gene should follow a negative binomial distribution. Therefore, we fit negative binomial distribution for every gene, regardless of whether the real data has one cell type or two cell types. We just try to do the maximum likelihood estimation to get a negative binomial estimated. And for the gene-gene correlation part, which is the joint distribution part, is the most important part because otherwise the data we simulate wouldn't be realistic. We use the joint modeling via the use of Gaussian copula to capture gene-gene correlations under the null model that there's only one cell type. So the reason we chose the Gaussian copula is because it can give us a homogeneous cell population after simulation. So specifically, we perform the following distributional transform first. So for every gene in the target data, we convert its counts to the cumulative distribution values. If you still recall from your probability class, the CDF values should be between 0 and 1. So after we convert every gene value counts into CDF values, we further go from the CDF values to standard Gaussian values. So taking the CDFs, we obtain the corresponding quantiles in the standard Gaussian distribution. So we get those two new sets of values. And with those values, to capture gene-gene correlations on this new scale, we fit a multivariate Gaussian distribution. That's it. So for this distribution, since marginally every gene is standard Gaussian with mean 0, variance 1, what's left for us to estimate is the correlation matrix, gene-gene correlation matrix in the multivariate Gaussian. With that estimated, the remaining is sampling. So we first sample from the multivariate Gaussian, and the sample we draw can be considered as synthetic cells. And the values for every gene are still in the Gaussian, standard Gaussian scale. So we go back, we convert the standard Gaussian values to CDF values, and using the fitted marginal negative binomial count distributions, finally, we convert the CDF values into counts. So that's why our synthetic cells would have counts, and as in the original scale of the target data. So I hope this explains the thought process of how we generate the synthetic null. And the homogeneity of synthetic cells is actually guaranteed by the multivariate Gaussian. So I want to quickly show you that this is how our synthetic null look like. This is how it would look like if you just do permutation. If you just randomly permute the genes, like we did in SED. So here, because we still want to preserve the overall topology of cells, like real cells. So our goal is more complicated than the original, the SED paper I just presented, because in that case, we just need to disrupt cell-cell relationships. Here, we need to have this null or negative control that mimic real data well enough. So benchmarking or doing the clustering followed by DE would give us a meaningful result, because otherwise it may not be a good contrast. So permutation doesn't give us a very reasonable synthetic null data, nor does the statistical literature. There's a method called knockoffs, and so the knockoff data also doesn't look like the real data. So then finally, my last question, question 3, is what's next? So assuming we have the synthetic null, how can we use it to reduce false discoveries? So for this, I want to propose this contrast strategy. So back to this example, single cell post-clustering DE analysis. So I've already talked about using SCD3 to generate synthetic null, right? Then we have target data parallel with synthetic null data. We can perform clustering, and you have the choice to pick a clustering algorithm you include in your pipeline. So you apply the same algorithm to target data and synthetic null data to obtain the clusters. Then for every gene, you perform the DE analysis between the 2 clusters. You can choose a statistical test for DE, like the Wilcoxon I mentioned, or you could choose other things included in the popular Surat package. So with this, for example, if you get a p-value from Wilcoxon, then you can take the negative log p-value to get a DE score. The reason is that a smaller p-value, the more likely the gene is DE. So therefore, this negative log p-value can be a reasonable DE score. So every gene, each of the M genes will get a DE score in target data and in the null data. Last step is to do a contrast, right? How do we do the contrast? For this, I will introduce our method previously published as Clipper for doing the contrast. And I will introduce this step in more detail, but together, this method we've developed is called cluster DE for addressing the double dipping problem. So Clipper is a strategy that follows the contrast to achieve p-value free FDR control. So it was led by my student Xin Zhou. It was published in 2021. So there are several advantages of using Clipper versus the traditional p-value based false discovery rate control. So first, Clipper doesn't require every gene in our case to have a high resolution p-values. It can just need a contrast score. So getting rid of this p-value requirement, we actually avoid parametric assumptions like in the negative binomial case for bulk RNA-seq. And also we don't need large sample sizes like in the Wilcoxon test to get high resolution p-values. Clipper has 2 components, contrast score, 1 per each feature. So in our case, a feature is a gene, and a cutoff on the contrast scores. So suppose that using our cluster DE strategy, we take the difference between each target DE score and the null DE score for every gene. We get a contrast score for every gene. And pooling the contrast score together, we get a distribution here. And the assumption in Clipper is that this distribution should be roughly symmetric about 0. The reason is that we believe the majority of the features or genes should be non-DE, should not be informative, should not reject the null. So therefore, they should be equally likely to be positive or negative. And the assumption is that target DE score or null DE score, which one is bigger, it will become a random thing for those null genes or non-DE genes. So with this assumption in mind, basically this cutoff is based on the statistical literature called knockoff. I mentioned it's a way to generate null data, even though it doesn't really work well for the single cell case, but it was developed for the high dimensional prediction model variable selection or feature selection problem. So it has a theory for finding a cutoff on the contrast scores to control the false discovery rate. So the intuition here is that we think for a threshold T by symmetry, the left tail, the red tail is false discoveries. The blue tail are all discoveries. So therefore their ratio represents false discovery rate. That's the intuition behind this formula. And our idea is to push this threshold to be as small as possible so we can maximize the number of discoveries and the theories from this statistical paper. But I want to say that this idea would only work well when M is large, when you have many, many features. So they themselves serve as a way to get this symmetry. Okay, so then the contrast score construction becomes the key part of using Clipper idea. So our target data is the actual data, synthetic null data is the negative control, right? So this allows us to get rid of the worry about how can we derive a null distribution for accurate p-value calculation. We don't need to worry about that. We just need to apply the same, however complex pipeline to the two data sets in parallel and do a contrast in the hope that for most of the features that are null features, the difference will be equally likely negative or positive. So that's the Clipper for the last part. And we use the strategy to find the cutoff, and the genes whose contrast scores are greater than the cutoff will be called DE genes. So last, I want to show how it works on real data. There are several expectations we expect a reasonable DE method to achieve in this problem. Expectation one is that no DE genes should be found from one cell line, because we know this is the case where no more cell types should be there. Every group of cells should just be one cell line. So in this case, you can see that our synthetic null data did mimic real data pretty well, and cluster DE did find no DE genes in most cases. In contrast, SURAT, which is the state-of-the-art pipeline, found thousands of genes as DE, sometimes even close to 100% of all genes, which is definitely not reasonable. Count split and TM test are two existing methods that were developed to counteract double dipping. But unfortunately, their assumption requires genes to be independent, which doesn't really hold well for real single-cell RNA-seq data. That's why they also have severe inflation in false discoveries. The second expectation is that when there are actually more than one cell type marker genes should be found by the DE analysis. So in this case, we look at two monocyte subtypes, CD14, CD16. They are biologically known to be different. In this case, our synthetic null still mimic real data. And you can see that we do find some genes as DE gene in this case. And one thing to note is that cluster DE, SURAT, count split can all allow any DE test. So that's why Wilcoxon, t-test, and so on, 5 tests in total are included here. And we can actually see that based on our empirical evidence, Wilcoxon performs the best in terms of the FDR control and also the consistency across replicates. So we pick Wilcoxon. And you can see that cluster DE did find some DE genes in this case, even though the existing methods still find thousands of genes in most of the cases, which is kind of not so different from the cell line case. So besides this expectation 2, expectation 3 is that housekeeping genes should not be found as top DE genes, right? So cell type marker genes should be in the top. Housekeeping genes, which are expressed everywhere, shouldn't be among the top because they are not useful markers for cell types. So therefore, we did show that in our cluster DE result, this well-known marker gene CD16 has a top rank, while a well-known housekeeping gene B2M has a low rank. So it has a very clear distinction. But in SURAT, they were ranked similarly, suggesting that if you don't have the knowledge, you look at SURAT results, you cannot distinguish the two. And also we look at the expression of top 5 DE genes found by cluster DE, and we did see that they have very distinct distributions in terms of expression levels in the two clusters, while the top DE genes found by SURAT have very similar distributions in the two clusters. Another evidence showing that they are not really indicative of cell types. Finally, we look at the so-called gene set enrichment analysis, a commonly performed analysis in bioinformatics, which we take 2 gene sets, a set of genes known as monocyte marker genes and a set of housekeeping genes. We check which positions they are in the ranked D gene list in cluster D and SURAT. The results to interpret the curves, it shows that the first set, marker genes are enriched in the top of the rank list in cluster D, but the housekeeping genes are enriched in the bottom. In SURAT, they were hardly distinguishable. So it's another evidence showing that we are better in distinguishing cell type markers and housekeeping genes. And finally, I want to ask this question. Why can cluster D remove housekeeping genes? This plot says why. So in the x-axis, we showed the average of the target D gene score and null D gene score for every gene. The y-axis shows the contrast score. So for SURAT, its cutoff or its D gene identification is only based on the target D score. That's why you see this ACB, B2M, they have pretty high target D score, and they will be found by SURAT. But we do the contrast score. We use the y-axis. Then you can see that after doing the contrast, their contrast scores are not so high, which means that target D score are not so much higher than null D score, which is also high. Therefore, they counteract each other and they will not have a high contrast score. So they will not be caught by cluster D. So finally, I want to just quickly say that I think this contrastive strategy is computationally efficient versus the traditional p-value based strategy. Even though I didn't talk about this work in this slide, but I want to say that my student Heather Zhou has implemented the strategy in the eQTL analysis and showed that compared to the state-of-the-art fast QTL method, we can find almost identical discoveries, but with the running time reduced by 100-fold. So I want to say this is a very useful strategy when you did have a lot of features to examine. This is our ongoing work. So finally, summary. I talked about 3 questions. First, what is a proper null hypothesis? Different null hypotheses may lead to different discoveries. And I show one example about bulk RNA-seq-DE analysis, MB test versus Wilcoxon test. And we show using permutation that there are a lot of false discoveries when the MB assumption doesn't hold. Second, how to make an abstract null hypothesis concrete. For this, I showed one simple example for detecting dubious t-SNE UMAP embeddings, and this is via the use of permutation, same as in example 1. But for the more complicated single cell clustering followed by DE analysis, I propose to use our simulator, SCD93, for generating the synthetic null. Last question, how to use synthetic null to reduce false discoveries? For this, I propose to use this contrast strategy by our method Clipper. So together, synthetic null and contrast strategy give us a solution to example 3. How do we do single cell post-clustering DE analysis? And that is our cluster DE method. So I want to say 2 more take-home messages before I wrap up my talk. The first message I want to say is that synthetic null can make an abstract null hypothesis concrete for our users, and it can enable contrastive data analysis. So based on my communications with data analysts in the field, I feel like statistics is usually something that considered to be mysterious, abstract, hard to interpret, right? So therefore, even it's already complicated, people go with popularity and choose the method that other people use. But the fact is that if you have your own data and popular method has its own assumptions, they may not match. And therefore, it may give you false discoveries. How do we counteract that? Generating synthetic null as some concrete object like your real data, so you can do an analysis in parallel. I think it's a way to make abstract statistics more interpretable and concrete. So I want to say this, there is a quote from a Chinese proverb called, teaching a man to fish is better than giving him a fish. 授人以渔不如授人以渔. So it sounds the same, but basically means that we should teach our analysts how to evaluate methods themselves, rather than just say, oh, go with the popular method. The second take-home message or question I want to say is, do we still believe less is more? It's a common saying in many places, but in our field, it's not always the case. First, less is more can be reflected in this Occam's razor principle, the principle of parsimony. if we have multiple solutions, we prefer the simpler solution. And we think that may reflect the scientific truth. However, as many of you know, nowadays, computational method development is going for more complicated methods instead of simpler methods for many reasons. But I feel like this trend is opposite to the outcomes razor. So therefore, I want to bring this up. Do we still think this is a reasonable principle? Second, I think fewer but more reliable discoveries can better lead us to science. But on the other hand, the reality is that in many cases, a method gets popular because it can give you many discoveries. Therefore, you can more easily make a story out of the many discoveries. But does that mean more reliable? Maybe not. So ultimately, I think this philosophical difference, or actually I should say the disciplinary difference, is science versus engineering. I think when we discuss a method choice, whether we go for a simpler method or more complicated method, we should think, do we really want to discover scientific principles, or do we just want to have a tool for use? I think this is something that needs a lot of us to think. Finally, I want to give acknowledgements to many people, starting from my PhD advisors at Berkeley. Without them, I couldn't be where I am. But Dr. Peter Bacon and Dr. Haiyan Huang for their unconditional support when I was a student who didn't know anything. And I want to thank my students, including Xinzhou, who will join Oregon State as a faculty member this fall, for developing Clipper, Heather for the Clipper QTL thing I briefly mentioned, and for the two simulators, Dongyuan and Tianyi led effort, and Qingyang also helped with the SEM3, and Guangao helped converting the simulator to generating reads. And for the cluster DE, Dongyang Kexin led effort, and Christy for SED. And I also want to say I have former trainees who ended up in academia after PhD training, which means that they still find passion in research, and I should be happy about. So Vivian and Nan. And also I want to thank my many collaborators, including Dr. Wei Li and his former postdoc Yu Mei Li and Dr. Lucy Xia for collaborating with me for the work I presented today. And also my other important collaborators, Mark Biggin, for leading me through many projects when I was a student and when I was a junior faculty. And also Xin Tong, Rachel Wang, and Yang Feng for collaborating with me on other classification projects, and Shi Hua at Chinese University of Science, Chinese Academy of Science, and also my colleague Arash at UCLA. So without their collaboration, I couldn't have done so much. And of course, the funding agencies for the support. Thank you.
That's a wonderful talk. Thank you so much, Dr. Li. Really enjoyed. I feel like I want to go back immediately, update all our data analysis pipelines, just a lot of work to do, a lot of update, and I'll take the privilege to ask the first question. Actually, right now, this moment, I don't want to learn how to fish. I want to get the fish immediately. So I want to kind of figure out the best pipeline, but in single cell differential gene expression analysis, we do, have these issues actually frequently, like low quality, high quality cells. Low quality in general, it have lower total counts. But with current popular methods after normalization, there's low quality cells tend to have extremely high expression levels among the much less genes that were detected. I just wondering, like, if we switched to SC93 combined with Clipper, would this issue got addressed?
I would say yes. I think there are a lot of work that needs to be done, but I think Clipper, it does provide a more flexible way for evaluating false positives. Because I think one thing you could do with Clipper, which I think make it intuitive, is this, checking the symmetry. So we have seen examples where the Clipper symmetry assumption doesn't hold well. So once you obtain this contrast score distribution, it doesn't look symmetric. Maybe the mode, the center is to the left or right of 0. And that can give you some information about whether the null, the synthetic null or real null data is something that can serve as a reasonable contrast. And I think we just made the first step in generating synthetic null data for single cell data analysis. And there's a long way to go because in single cell data analysis, we know there are more than 1,000 methods, right? Computational methods that can discover a lot of things that couldn't be verified from data. Then in this case, experimental negative control is clearly not possible. And positive control is also very difficult. If we can get positive control, it means we know the scientific principle. Then what do we do? So I would say even the positive control is very difficult, but negative control, just like the thing we get from permutation, seems to be feasible and more feasible to me. So that's why I would like to invite any thoughts from the community so we could help generating synthetic null under more scenarios besides this post-clustering DE problem.
Yeah, very true, very cool.
Yeah.
So yeah, thank you, Dr. Lee. So let's go over the questions in a QA. Are those visible to you? Or I can read them one by one.
Yeah, I can look at them. I can see all of them. Yeah.
Yeah, you probably can read the question and then go through them one by one.
The first one, why permutation cannot give a perfect null control compared with SC design 3? So depending on the question, depending on context, as in SCD, we found permutation is good enough because in SCD, the null just requires us to disrupt any cell-cell biologically meaningful relationships. That's it, and permutation can do the job. And I didn't say that in SCD later, we added a comparison by using SC design 3 as the null control. So we use SC design 3 to generate the cells, assuming the cells are homogeneous. But in that case, the good thing is that the null distribution of reliability score, the thing we define, doesn't change much from going from permutation to SC design 3. So that's a good thing. But in the clustering followed by DE problem, it's a more complicated problem because you need the cluster, you really need a cluster to have some reasonable matches between target data and the null data. So therefore, if the cells are generated by permutation, by randomly permute each gene, then the cells would have I should say in the synthetic null data, the genes would have no correlation at all. And then the data would not look like the real data. So then if you do clustering, the resulting clusters found from this permuted data wouldn't really correspond well to the clusters you found from real data. That's the problem. And therefore, it's not as reasonable null control as the SDN3 null control, at least based on our intuition, it should be the case. But of course, I would say this is something we welcome comments, criticism about whether there's a more reasonable approach. And for the second question, how well can SCD deal with data with different batches? If you run a data with harmony for batch collection, do you still compare UMAP with PCA to calculate reliability score? I think it's a different question because harmony will basically project batches, two batches to the same UMAP space, for example, and then you will have the UMAP embedding. But in this case, your pre-embedding space doesn't have batch effect removed. So it's not a thing we're evaluating. We're just evaluating the projection, embedding step. We're not evaluating the integration step. So whatever integration algorithm you use, to use SCD, you need to have pre-embedding neighbors and post-embedding neighbors that should be the same if the embedding does a good job. But for the integration problem, in the pre-embedding space, your two batches are not mixed, then they are only mixed in the post-embedding space. So in this case, you cannot really evaluate the neighborhood consistency before and after embedding. So that's not a case where SCD can evaluate. Yeah, unless the integration method can give you pre-embedding integration result, and then you do UMAP t-SNE after the integration. That's the thing you can do SCD on, but it's not for harmony. Okay, I hope I answered this question. Next one, cluster DE reduces the false discovery in the single cell line population compared to the other method, but did it also discover some DGs that might be real, as in we know even a cell line can have heterogeneity? That's a question I got from another audience when I gave the talk. And I really want to clarify this. A cell line a cell line has cells with variation doesn't mean there are two cell types. That's the thing. So basically, the application of cluster DE is to review new cell types and their marker genes. But why would you believe that the heterogeneity or whatever you see a cloud in the one cell line should indicate there are two cell types? I don't think so. So I think the within cell type heterogeneity is not what the signal cluster D is looking for. Cluster D is looking for only the signal that distinguishes two cell types. So that's why we use single cell line data as the negative case to show that no DE genes should have been found. So I hope this answered the question. But it's a very good question. The reason is that it's an open question in the single cell field, in my opinion, what the within cell type heterogeneity really means. How much is biological? How much is technical? They're actually mixed together. Yeah. Okay. And there is a question about why I called the null a synthetic null. I understand you don't care about causal inference, but just association. That's fine. But in the end, you are proposing an estimator. And essentially, I see what you do as proposing a synthetic estimator, or in other words, a synthetic control variable. Could you maybe clarify? Is it fair to say what you do is to propose a synthetic estimator under the same null? Okay. I want to clarify what I did is that I have a null hypothesis and a null model. I used real data to estimate this null model parameters. So given this model parameters estimated, I generate data, I simulate data by sampling from this fitted null model. So in other words, a better analogy, which I should probably add to my slides, is that if you have heard of likelihood ratio test, what we did is very similar along the line, because in likelihood ratio test, you fit a model, you find the likelihood, you maximize the likelihood under the null hypothesis, under the alternative hypothesis, you compare these two likelihoods to see which one, whether the alternative likelihood is much bigger than the null one to a great degree, then you reject the null. So that's what the likelihood ratio test is doing. What I did here is also to fit a distribution as in the likelihood ratio test under the null hypothesis. Given the distribution fitted, I sample from the distribution and I call that synthetic null. Okay. Since the synthetic null is randomly generated, do we need to do multiple replicates of it too? We have a result which I didn't include, but we actually show that yes, this contrastive strategy can accommodate one synthetic null, two synthetic null, or any number of synthetic null. So ultimately, it is a choice of computational resource and the stability you want to achieve. The more you generate, the more stable the result. But actually, I should also say the FDR control theory applies on average. So which means that it's an expectation. It's controlling the false discovery rate, which is itself an expectation. So anything it can say is about the average across many uses of the same procedure. It's not about one repetition. So that's why a random procedure like we did can still control the FDR, even though I agree, using different random seeds, you will get a different synthetic null and you have some different randomness, and then the results will be slightly altered. But we do have the stability analysis in our paper to show that the stability is generally good enough for making discoveries. And also like permutation has the same thing, right? How many number of permutations you want to do to get the result stable? I think it's the same thing. And is there a tutorial for integrating the usage of these tools for single nucleus RNA-seq multi-ohm data? My interpretation of the question is, we do have tutorials for each tool I discuss here, because I basically mentioned, how many? I mentioned three tools, right? Or four tools, SED, Clipper, SED3, and Cluster DE. We do have tutorials, but regarding multi-ohm data, scRNA-seq data, single nucleus, I would say for single nucleus RNA-seq data, I don't think the analysis should be different from single cell RNA-seq data if you do differential expression analysis for each gene. For multi-omic data, it's more tricky because you need to define features for the attack-seq part. But once you have the features defined, the post-clustering DE problem should be the same formulation. But maybe you need a different distribution for the synthetic and null. And in SCD93, we do have a way to generate single cell ATAC-seq, but it's every feature or peak, the marginal distribution is no longer negative binomial. So yes, I would say, hopefully we will make a place to have things listed in one place, but it's something we have in mind. And how can we integrate the tools in one place so people have one tutorial to follow like Surat? Probably that's the thing we need to spend effort on. But so far, every tool has its own tutorial. And Linda, and thank you for the talk. So for Clipper, I still don't quite understand why it doesn't need to rely on high resolution p-value. Could you explain it a bit more? Thank you. The reason is because it uses a different theory for FDR control. So p-value FDR control requires p-values to be uniformly distributed between 0 and 1 under the null. That assumption is replaced by the symmetry assumption we have for contrast score here. So I'm not saying that the traditional p-value thing wouldn't work if you have low resolution p-values. So in the extreme case, your p-value could be binary, 0 and 1 p-value. So if under the null, the 0 p-value and 1 p-value are equally likely, the traditional FDR control, Benjamin Hochberg, can still control the FDR, but you will have almost 0 power. So you will lose the power at the sacrifice of FDR. So in that case, the high resolution was actually granted by the resolution of p-value, given the p-value is valid. In Clipper, the reason why I emphasize this N must be large is because you are using this multiple tests, many tests all together to serve as its own contrast. So here the thing is that if you have many tests and each test, or I should say the majority of tests should be from the null, then the contrast score should be symmetric about 0. And this is a way to give you a good estimate of the tail probabilities, the red tail and the blue tail, so you can have a good estimate for FDR. That's why you can control it well. So that's why here the M must be large is the necessity. And the reason we have many contrast scores to serve as its own distribution contrast, we don't need high resolution p-values. I don't know if this makes sense. So it's the power of many tests. Okay, is the perplex, finally, the last question, is the perplexity parameter for 2D embedding based on different distances? Could you provide a PPT for us? For the first question, based on different, I don't understand why the perplexity parameter is based on different distances. It's just the t-SNE perplexity. So it's a t-SNE hyperparameter in its own algorithm. And if you ask about the distance we use for SCD, that's a different question. We basically evaluate this Euclidean distance between every cell and its neighboring cells in the 2D space. So we have 2 sets of neighbors, one set based on the 2D space, another set based on the pre-embedding space. And then for each set, we calculate the Euclidean distance in the 2D space between each neighbor and the cell we look at. So this is distance we define. But the perplexity is t-SNE's own parameter. So for that optimization step, we just do grid search. We try multiple perplexity, run SCD, count the number of dubious cell embeddings, and then we pick the perplexity that gave us the smallest dubious cell embedding number. That's what we did. So yeah, so basically the perplexity is the built-in perplexity. We didn't change anything within t-SNE. And for the PowerPoint, I don't mind sharing. So if Dr. Yeah, so if you want to ask for a copy, you can email me and I can share it on my website.
Wonderful, very cool. So congratulations again, Dr. Lee, Professor Lee. They talk like to Professor Lee. Okay, so yeah, congratulations again. And I think the presentation is wonderful. I really enjoyed, and the QAs. Thanks for all the wonderful questions and the perfect answers. Great, awesome. I really enjoyed the night over here, and it's morning, Saturday morning in China. So thanks a lot. Thank you, everyone.
Thank you so much, Rudy, and thank you everyone for attending my talk on Friday night and Saturday morning. Thank you.
Cool. Bye.
Bye. Thank you.